My name is Pilar Rivero-Ríos from the University of Michigan, and I'm going to present my JCB paper entitled Recruitment of the SNX17-Retriever Recycling Pathway Regulates Synaptic Function and Plasticity. SNX17 defines a major recycling pathway which is responsible for the recycling from endosomes to the plasma membrane of several cell surface proteins. This pathway is composed of SNX17 and 3 different protein complexes, which are the retriever, CCC, and wash complex. Importantly, there are a series of alterations or mutations in components of this pathway that are related to neurological syndromes, including Down syndrome, X-linked intellectual disability, 3C syndrome, and Alzheimer's disease. But surprisingly, little was known about the roles of SNX17 in neurons. By analyzing the colocalization of SNX17 with specific synaptic markers, we found that this pathway is abundant at synapse. And around 50% of excitatory synapse contains components of the SNX17 pathway, which made us wonder what is SNX17 doing at synapse. To answer this question, we performed electrophysiology and measured miniature excitatory postsynaptic currents. We found that when we decrease the levels of SNX17, there is a decrease in the frequency with no changes in the amplitude of miniature excitatory postsynaptic currents. And the reason for this is a decrease in the number of dendritic spines, which are the places where most excitatory synapses occur. We also identified a role for the SNX17 pathway in long-lasting synaptic plasticity. We added a chemical stimulus to promote long-term potentiation, and this has 2 effects. There is a potentiation of the synaptic currents and also an increase in the size of dendritic spines. However, when we decrease the levels of SNX17, these 2 changes do not occur. We also found that SNX17 is actively recruited to dendritic spines upon long-term potentiation. And this recruitment is dependent on the binding to the retriever because a SNX17 mutant that is unable to bind the retriever does not get recruited to spines. Finally, we wonder which surface proteins are responsible for the synaptic roles of SNX17. And by surface biotinylation assays, we identified beta-1 integrin as a SNX17 cargo in neurons. We found that when we perform long-term potentiation, there is an increase specifically in the surface levels of beta-1 integrin, and this is completely blocked upon SNX17 knockdown, which suggests that the synaptic roles of SNX17 are mediated at least in part by beta-1 integrin. And if you want to know more about this paper, please visit the JCB website. Thank you for watching this video.